Hello, my name is Miguel Jimenez. I'm a assistant professor in biomedical engineering, and today I want to tell you how we're applying microbial devices to fertility hormone monitoring. As you may know, reproductive health monitoring unduly burdens women because of the need for in-person blood draws. This is particularly an issue for women undergoing assisted reproductive technologies because of the required sort of repeat samples to track ovulation and early pregnancy. You may know that there's sort of a range of at-home kits that can be used for testing hormones in urine, but as it turns out, ultimately, those must be followed up by in-person blood draws because these at-home kits do not provide sort of the quantitative results required by doctors to track fertility. So you might ask, why is it that we can't do an at-home blood test, sort of much the same way we use blood glucose monitors, sort of in patients with diabetes? As it turns out, the technical reason why this is not possible is because the detection method that is used today is incompatible with whole blood and therefore sort of requires trained personnel and analytical equipment that takes up sort of like a whole room to actually get that quantitative result. And so what we are doing is developing microbial devices, which are these sort of miniaturized systems that have the capacity to directly quantify molecules in complex samples. And so you might ask, sort of, what are these microbial devices? And so these are sort of blueberry-sized systems that integrate electronics with living whole cells to detect molecules directly. So as you can see here on the right side, the way they work is you can put a sample on the top, and that sample diffuses into the device, and then these onboard living microbial cells detect the target molecule and deliver a response, and that response is converted to a wireless signal by custom wireless electronics developed by our collaborator Rabia Sisigi in the electrical engineering department. And so together, this integrated system converts the levels of a target molecule into a wireless signal that might be read out sort of on your personal device. And sort of the key advance of these systems is really leveraging the versatility of living microbial cells. As you may know, microbial cells are sort of found everywhere in sort of crazy places, sort of on our bodies, inside us, in the soil, everywhere. And so sort of our thought is to sort of leverage that versatility of these cells to function directly in whole blood to avoid the need for that sample processing. And so with some early guidance by one of our later speakers, Dr. Kuhong, we're now applying this technology for monitoring fertility hormones in whole blood. And we've just sort of we've just gotten started with this, and some sort of early results by some of our graduate students and undergraduates here on the right side, we can see that, in fact, we are able to see some early signs of function of some of these sensor cells directly in whole blood. And so, as you can see, those signals are still low, so there's still a lot of work to do, but we hope that we can eventually apply this technology to reduce the burden of women by reducing the number of in-person blood draws that are required to sort of track fertility and reproductive health. Thank you.